Great, thank you very much. It's going to be a little bit of a challenge to follow up that excellent presentation, but I will do my best. Can you guys see my screen?
Yes, we can.
Okay, great. So it's a little bit of a pivot. My name is Alex Butler. I'm a medical student at Columbia, and I've been working with Dr. Chunhua Weng in the Department of Biomedical Informatics. And we're going to be talking about facilitating COVID-19 clinical trial search, analysis, and collaboration. As many of us are I'm sure aware, clinical trials really are the gold standard for generating high quality medical knowledge. And in the times of COVID-19, they're ever more important. They're coming at an unbelievable rate. As of yesterday, putting these slides together, there are more than 2,100 trials across the world and over 450 in the United States alone. You can cause a number of significant recruitment challenges in finding participants. And unfortunately, though we like to use machines, we like to use complex programs to help with this process, much of the data exists largely in free text and is not amenable for computation. And there are a lot of research questions we want to ask about COVID-19 clinical trials. Who's eligible for these trials? And is this representative of the patients we're seeing? Have the designs, have the eligibility criteria, have the treatments for these trials evolved over time? Can we identify opportunities for researchers to collaborate on these studies? And then finally, can we help patients and clinicians find COVID-19 trials? So out of Dr. Weng's lab, really what we've been trying to do is develop a machine-readable COVID-19 trial dataset to enable three primary goals. The first is to allow patients to find suitable trials. The second is to allow clinicians to find experimental therapies. And third is to allow investigators to reuse this knowledge for trial design and for collaboration. What this ends up is this very large flow chart. I know it's a lot of stuff to look at very quickly, but it's what I'm going to walk through today. The first is what Dr. Weng's lab has a lot of experience in, is making sense of clinical trial information, specifically eligibility criteria, basically the rules that a patient has to meet to go onto a study. So we download information from clinicaltrials.gov, do a significant amount of retrieval and annotation to create a structured database. We also work with a data set called AACT. It's provided by a group out of Duke University called Clinical Trials Transformation Initiative, which is incredibly helpful. From this data, we can do a significant amount of trial analysis, which I'll be very briefly highlighting here. We have a number of projects going. So if anyone has any questions, please feel free to ask them here, or if you want to reach out to me after the symposium, I can try to answer a little bit more thoroughly and in more detail. And then finally, with this data, we try to make it as accessible to as many people as we can. We've developed the COVID-19 trial finder to meet these three aims that I mentioned before. I'm going to hit on this at the end of the presentation. Very quickly, it's important to highlight what eligibility criteria are. Like I said, they're basically rules that every patient has to meet to go into a clinical trial. Right now, they're stored primarily as free text. So here's an example of just 2 criteria here. Some can have as many as 50 or 60 eligibility criteria. And because they are often very complicated, have a lot of medical terminology, it's very challenging for a computer to make sense of them. So through a combination of automated methods, as well as some manual review, what we have to do in our lab is convert it into a structure more similar to this. Now what you're seeing is we've actually annotated these criteria. So we tell the computer what pieces or words should be considered as important. Are they a disease? Is it a measurement? Is it, let's say, a lab value? And then how do these relate to each other? This allows us to really make sense of these criteria on a large scale. So our database right now contains about 415 COVID-19 trials across the US, falling into somewhere about 440 patient cohorts. For all these studies, about 69% of them are interventional, 28 are observational, with 3% being expanded access for various drugs. And on the right, we can see the drugs being used currently in the phase 3 or phase 4 studies. The peak is hydroxychloroquine, which has about 25 studies going on in the US, followed by other drugs we hear about a lot, is azithromycin, tocilizumab, remdesivir, and convalescent plasma. By breaking down all this information, we're also able to get a sense of the status of the patients that they're trying to recruit. So 64% of the trials are looking for patients who have confirmed COVID-19 diagnosis. 10% are actually looking for those who have, let's say, no exposure, no confirmed diagnosis, perhaps healthy volunteers. And 3% are looking for patients who've had the disease and have actually already cleared it. In terms of exposure status, we also see 11% of studies looking for particularly high-risk patients. This is usually defined as known exposure or healthcare worker, so it's another huge area for these studies. We also collect a little bit of information about hospitalization. So it turns out about 44% of studies actually require hospitalization, or 11% excluded, suggesting there's a really large group of these studies looking for hospitalized COVID-19 patients. And finally, on the right, we see that there's usually a time frame for when they were diagnosed to when they're eligible for the study, and this is a plotting of this days to diagnosis. And with this data, we're also able to plot on a map where these trials are popping up. So this is a heat map of the United States of interventional COVID-19 clinical trials. As you might expect, many of the hotspots surround big cities. So we're looking at New York, around Boston, Chicago, and a couple of big cities in Texas as well. This is true of the West Coast. We see Los Angeles as a big hotspot, San Jose and Seattle. But unfortunately, there are large areas, especially in the Midwest, that don't necessarily have the same trial availability. So for example, Wyoming, North Dakota, in this map have no trials looking for patients and can oftentimes be very far apart. So hopefully with this kind of information, we're hoping to match this with real patient data, identify areas where clinical trials should open. We're also with this able to track which drugs are being used. So we can see the growth in hydroxychloroquine studies, azithromycin studies, and remdesivir studies over time. And leveraging eligibility criteria data, we're able to make sense of how are we looking for patients for these studies. So just as an example, we've done a little bit of clustering analysis for clinical trials using hydroxychloroquine. And we ended up with a number of different groups of these eligibility criteria. Group 6 is one of the ones we pulled out, seems to target kidney disease. So the two concepts we're looking at here is dialysis and chronic kidney disease, or CKD, and we can see that they change in a very similar trend over time, starting very commonly being used and then drop off a little bit, ending around 45% in the current trials. Another group that we've pulled out, and I'm sorry, it's very hard to see these names. 4 of them are 4 drugs, so procainamide, flecainide, propafenone, digoxin, and finally long QT syndrome seem to relate to arrhythmia. So we know that hydroxychloroquine clinically, has a risk of causing de novo arrhythmias in patients. And we can actually see these eligibility criteria being used around the same time, coming up to a peak and now resting again around 40%. Another one of our research efforts is actually looking at criteria competition. So how are clinical trials competing over patient populations? We've completed a proof of concept. So we'd be able to calculate similarity between all the trials currently in the US. Starting right now with the criteria concepts. So what are they actually screening for? And then we can group these studies by zip code. So if I wanted to find clinical trials in New York, I could not only find out where they were, but I could see how are they competing for similar patients. The next step for this is actually to improve its accuracy. We want to be able for all these studies with a given COVID-19 patient data set actually identify the eligible patients so we can directly measure the overlap of target patients. So allow us to measure competition, but also to find patients who may be not totally available for the clinical trials that are available. Another large area we're getting into now is actually finding opportunities for collaboration. So researchers, as a number of journals have pointed out, researchers are struggling as case counts fall. Falling COVID case count is excellent news, but clearly with so many trials opening and so many looking for patients, it can cause a lot of troubles. Now, there have been some efforts actually hoping to foster this kind of collaboration, whether it's sharing data, merging studies. One example out of Johns Hopkins is covidcp.org. So in our lab, we thought maybe we can use our structured data to try to identify similar trials for collaboration, both with the structured data, free text information, as well as location of the recruiting sites. And essentially for research coordinators, for research investigators, we can recommend similar trials that they may be willing to work with. Very briefly, this is just a sample of the output that we've been able to generate. So each of these graphs relates to a single COVID-19 clinical trial, and each bar is another clinical trial, essentially ranked in terms of its similarity. So we're going to take a look just at one quick example down here on the bottom left. Now, this study is essentially looking at convalescent plasma for the treatment of COVID-19. And we don't have to dive into this data in explicit detail, but this is really what's happening in the back end. The computer is taking a lot of the structured data we're able to generate, comparing it to the data for all the other trials in the data set, and coming up with what we're calling a similarity score, or a collaboration score. Essentially, how similar are these studies and how well could they collaborate together? It's very hard to make sense of this just with NCT IDs, but if we were to just look at this top hit for the most similar trial, that trial is titled Convalescent Plasma as Treatment for Hospitalized Subjects with COVID-19. So clearly they have the same intervention. A number of their other design characteristics are the same. So we see phase, study design, masking, start date are very similar. So there's a lot of similarities. And again, the next one in the line is another convalescent plasma study. All of these are generated completely automatically. None of us are sitting down and mapping these things out. So this is, we think, a really good step for helping to foster collaboration really across the entire United States. And then finally, one of the biggest projects that we've come up with in the last few weeks is the COVID-19 Trial Finder. Really what this does is allow users to search all COVID-19 clinical trials in the US. They can search by zip code and mile radius. They can filter by trial type, and they can search by keyword. But really where our site goes a little bit further is leveraging this eligibility criteria information. So we allow users to filter the studies by age, COVID-19 status, pregnancy, and then they're actually able to pre-screen themselves for these studies using dynamically generated medical questions. Now I have a short video I'd like to share. I'm just going to stop sharing for a second because I have to make sure that my screen audio is being shared. So give me 1 second, I'll jump right back into it. So this is actually the website that's currently being used out there. COVID-19 Trial Finder, like I mentioned, allows anyone to search for clinical trials. If, for example, I'm looking for studies maybe around New York, I can enter my zip code. There are some additional search parameters for more advanced users, potentially for research investigators or coordinators. I can look at keywords, I can look at trial type. But just by searching in the US, or around New York, excuse me, there are 67 nearby and active trials. I can then filter down by my age. I can say whether or not I've been diagnosed for COVID-19. Let's say I have not. I'm not currently hospitalized, not currently pregnant. And again, now I've been able to cut that 67 down into 18. Here are all the trials that I'm currently eligible for in my area. I can see any of their information pulled directly from clinicaltrials.gov. And if I would like to filter this list down even further, this website generates medical questions, again, based on the structured data we've been able to generate, that I can answer and actually pre-screen myself for these studies. So by answering these questions, I'll cut down this list of eligible trials even more. So now I'm already down to 13. So to just quickly summarize some of the efforts I've touched on. In Dr. Weng's lab, we've tried to generate a structured data set of trial features and eligibility criteria. We have a number of research projects going on and are always excited to collaborate on new ideas. And recently we've come up with this online tool for COVID-19 trial search containing all clinical trials in COVID-19 across the United States. This allows people to find studies by answering dynamically generated medical questions. And expansions in the future include searching by a word cloud, an actual map module to find recruiting sites, and also a summary of the data on the homepage so anybody can get a sense of what these trials look like. And then finally, we're also working on recommending similar trials to research coordinators, to research investigators, to help boost these clinical trials. Well, that is perfect timing because that is the end of all I have prepared. So I'm happy to take any questions from anyone who's listening.
So, Alex, have you seen, so how much utilization of this resource have you seen?
Yes, so we've been only sharing right now around, basically around Columbia, and are hoping now to start spreading out to more community engagement. I believe over the last 2 weeks, we've had a little bit north of 300 or 350 people come and use the site. Our also hope is that as more and more people use it and screen for clinical trials, we're capturing some of those filtering questions, so the average age, we're capturing information like COVID-19 status, what kind of studies they're looking for. So hopefully that information will also be able to inform other researchers and other coordinators on what kind of studies should they be opening for, or what kind of studies are patients looking for.
Yeah, and so there are also a variety of additional parameters that can be used to potentially judge the quality of a study. So I think there's a lot of futile studies that are actually subtracting patients and potentially exposing them to risk just because everybody is trying whatever their favorite drug is, and they're just very easy to open a clinical trial. Just don't need much background information. So, so is there any kind of information on the sort of the scientific background that led to the study, and whether there are other studies like this that already, for instance, provided some preliminary results on the futility or value of the approach?
Yeah, so I think you pointed out a very good point. It's very easy to get many of these clinical trials started. Unfortunately, with the data we're capturing, there isn't always a ton of, let's say, scientific background or scientific justification for the study. But one of the things we are doing is, in addition to just recommending clinical trials, we're also grouping them. So let's say for a given drug or for a given drug class, we can actually pull these trials together into one cluster, one small data set. And we're working with a number of members in the biostatistics department. They obviously have much more experience than we do in getting these studies started, in what makes a feasible study, and they may be able to catch some of these that say, this isn't as reliable, this isn't a study I would like to focus on. These are definitely much more a higher priority. So that's something we're hoping to do with this recommendation or clustering program going forward.